Welcome to Chapter 1, Learning About Life. What is biology? Biology is the study of life. Biologists may study anything from microscopic or submicroscopic views of a cell to the ecosystems and the whole living planet. We can define science from the Latin scientia, meaning knowledge, as knowledge that covers the general truths or the operations of general laws, especially when they are acquired and tested using the scientific method. The scientific method is a method of research with defined steps that include experiments and careful observation. The scientific method is the testing of hypotheses by the means of repeatable experiments. A hypothesis is a suggested explanation for an event which one can test. Natural sciences includes both life science and physical science. Life sciences are the sciences that study living things, for example, biology. The physical sciences study non-living matter, for example, astronomy, geology, physics, and chemistry. There are also many interdisciplinary sciences, such as biophysics and biochemistry, that build on both life and physical sciences. There are two main thoughts in scientific reasoning. One is inductive reasoning. The other is deductive reasoning. Inductive reasoning is a form of logical thinking that uses related observations to arrive at a general conclusion. Sometimes this is described as descriptive or discovery science. Its aim is to observe, explore, and discover. In deductive reasoning, this is a form of logical thinking that uses a general principle or scientific law to predict specific results. It is a hypothesis-based science, and it begins with a specific question or problem and has a potential answer or solution that one can test. Here's a flow chart of a scientific method. It begins with observation. Then we ask a question and propose a hypothesis. Then we test the hypothesis and analyze those results. If our hypothesis is supported by the data, then we report the results to the scientific community. If the hypothesis is not supported, it's best to rethink the hypothesis and try again. So, what makes a good hypothesis? A valid hypothesis must be both testable and falsifiable. It must be subject to experimentation, and those experiments may result in disproving the hypothesis. In testing, usually there is a treatment group and a control group. The difference between the treatment and control group is the variable. A variable is going to be the part of the experiment that is varied between the two groups. The control group contains every feature of the experimental group, except that it is not given the manipulation that the researcher hypothesizes. For example, here we have two groups, a control group and a treatment group, in a clinical trial to determine the effectiveness of a new antidepressant. In the treatment group takes one actual antidepressant tablet every morning. The control group takes one placebo every morning. Participants in either group do not know whether they are getting a placebo or the actual antidepressant. This is done to make sure that any difference between the two groups is likely due to the presence of the antidepressant alone, and not some other confounding factor, such as the placebo effect. Most scientists will present their results in peer-reviewed manuscripts that are published in scientific journals. Peer-reviewed manuscripts are scientific papers that scientists' colleagues review. Scientists publish their work so that other scientists can reproduce the experiments under similar or different conditions in order to expand on the original findings. The structure of a typical scientific paper can be abbreviated with the acronym IMRAD. The I stands for Introduction. The M stands for Materials and Methods. The R stands for Results. And the D stands for Discussion. However, there are also usually an abstract, acknowledgments, and a reference section. The abstract is a concise summary that exists at the beginning of the paper. The introduction should be brief but broad and have plenty of background information. It gives a rationale and justification of the work done. In Materials and Methods, a complete and accurate description of the substances the researchers used and the methods and techniques that were used to gather the data should be reported. In the results, there is a narrative of findings without any further interpretation. In the discussion, researchers interpret the results and describe how variables may be related. It is here that the author will attempt to explain the observations. Finally, we have the conclusion. The conclusion summarizes the importance of the experimental findings. So, what is life? Biology is a science that studies life. But how do we define what is living and what is not? At first glance, it might sound like a silly question, but it's actually not that easy to define. Scientists have put together a certain criteria for what is considered to be living. These properties include order, sensitivity or response to the environment, reproduction, adaptation, growth and development, regulation and homeostasis, energy processing, and evolution. In order, all organisms are composed of one or more cells. For single-celled organisms, atoms come together to form molecules inside the cell. These molecules then come together to form the cell's individual organelles. In multicellular organisms, similar types of cells come together to form tissues. Different tissue types come together to create organs, which are body structures with a distinct function. Organs will then work together to form organ systems, and those organ systems will coordinate with one another for the functioning of the entire organism. Organisms can respond to diverse stimuli. For example, plants can bend toward a source of light, or climb on fences and walls, or even respond to physical touch. Bacteria can move toward or away chemicals in a process called chemotaxis, or they can move toward or away from light in a process called phototaxis. A movement toward the stimulus is considered a positive response, while movement away from a stimulus is called a negative response. Single-celled organisms reproduce by first duplicating their DNA, and then dividing it equally as the cell prepares to divide, forming two new cells. Multicellular organisms often reproduce using specialized reproductive cells, called gametes, which are the oocytes or sperm cells. After fertilization, the fusion of the oocyte and sperm cell creates a new individual. All living organisms exhibit adaptation or fitting into their environment. Adaptation is a consequence of evolution by natural selection. For example, we have heat-resistant archaea that actually live in the boiling hot springs in Yellowstone. Another example is the long tongue length of a nectar-feeding moth that matches the size of the flower from which it feeds. Adaptations enhance the reproductive potential of the individuals that exhibit them. This increases their ability to survive and reproduce. Adaptations are not constant. As an environment changes, natural selection causes the characteristics of the individuals in a population to track those changes. For example, in the image, we see a blind cavefish. These cavefish have lost their eyesight and their eyes over many generations of being in complete and total darkness. Organisms grow and develop as a result of genes providing specific instructions that will direct cellular growth and development. Regulation or homeostasis refers to the relatively stable internal environment required to maintain life. In order to function properly, cells require appropriate conditions such as proper temperature, pH, and appropriate concentrations of diverse chemicals. These conditions, however, may change from one moment to the next. Organisms are able to maintain homeostatic internal conditions within a relatively narrow range, almost constantly, despite environmental changes. This is done by the activation of regulatory mechanisms. Living things are highly organized and structured, following a hierarchy that we can imagine on a scale from small to large. The atom is the smallest and most fundamental unit of all matter. An atom retains all of the properties of the element. It consists of a nucleus surrounded by electrons. Atoms come together to form molecules. A molecule is a chemical structure consisting of at least two atoms held together by one or more chemical bonds. Many molecules that are biologically significant are considered macromolecules, meaning large molecules. Macromolecules are typically formed through a process called polymerization. Polymerization forms a polymer. A polymer is a large molecule that is made by combining smaller units called monomers together. The cell itself is the smallest fundamental unit of structure and function in living organisms. Some organisms will consist of only a single cell, while others are multicellular. Scientists classify cells as either prokaryotic or eukaryotic. Prokaryotes are single-celled or colonial organisms that do not have membrane-bound nuclei. Eukaryotes are organisms that do have membrane-bound organelles and have membrane-bound nuclei. In multicellular organisms, similar types of cells come together to form tissues. Tissues are groups of similar cells carrying out similar or related functions. For example, adipocytes come together forming adipose tissue. Epithelial cells come together forming epithelial tissue. Muscle cells come together forming muscle tissue. And neurons come together to form nervous tissue. Organs are collections of tissues grouped together, performing a common function. Organs are present not only in animals, but in plants as well. Some examples of organs are the brain, the eye, the teeth, the skin, the heart, the lungs, and the stomach. An organ system is a higher level of organization that consists of functionally related organs. Mammals have many organ systems. For example, the circulatory system functions to transport blood throughout the body and to and from the lungs. It includes organs such as the heart and the blood vessels. An organism is an individual living entity. For example, in a forest, each tree in that forest is considered an organism. Single-celled prokaryotes and single-celled eukaryotes are also considered organisms, sometimes called microorganisms because they are so small. Biologists collectively call all of the individuals of a species living within a certain area a population. For example, a forest may include many pine trees, which represent the population of pine trees in that particular forest. Different populations can live in the same area. For example, the forest with the pine trees can also include populations of flowering plants, insects, and microbial populations. A community is the sum of all of the populations that inhabit that particular area. For example, all of the trees, flowers, insects, and other populations in a forest form the forest's community. The forest itself is considered an ecosystem. An ecosystem consists of all of the living things and non-living things in a particular area. The non-living parts of the ecosystem include things like nitrogen in the soil or rainwater. At the highest level of organization, we have the biosphere. The biosphere is a collection of all ecosystems, and it represents the zones of all life on Earth. It includes land, water, and even the lower atmosphere. Thank you for watching.